drawing assignment, and we'll go through the whole video. It's all covered in the book as well, but I organize it in a way that I hope is a helpful framework to try to help show you that we can build a framework for biology. We can see it in the textbook, and their textbook authors trying to do that too, but I just don't think textbook authors do a very good job with what I think is a central feature of being a biologist, and that is to think like a biologist, and that is to frame things, to say, well, what are the key highlights? How do we organize this knowledge in a way that's memorable? And so we're going to talk about 3 types of stem cells, as does our book. And we're going to roll through these, and at certain points I'll say, and we're going to draw this for our assignment. And I think there'll be 3 drawings that we'll do, and that we'll talk about them as we go through. And, you're just going to send me pictures of your drawings like that, okay? So here we go. They're really in nature. There are really 2 types of stem cells that we could talk about. This is an animal chapter. I should mention that there are stem cells in plants. So this is about animals, and the book is all about focused on animals. It doesn't even mention or even isn't thinking about plants. And our book is like that. Our book does not think about plants. It's all about animals, and our course has been almost all about animals. So it's an animal story for now, but there are parallels in plants. The little growing bud or the growing flower in a plant is stem cells. So it's pretty fascinating to connect that too. And but that's that. Then this third example is stem cells that are normal cells that get converted in a research laboratory or a clinical setting into stem cells. So 3 categories that we're going to talk about. And our favorite example for the first is muscle satellite stem cells. And what we want to say right away with adult stem cells is that's what they're called. Nobody's going to change the name of it, but it's a misnomer because satellite cells of the category that we're talking about are found in the fetus and even the embryo, and then child, teen, adult. They're really there for our whole life. And so they're called adult stem cells. That's not going to change. And there are specific examples scattered in different locations through our body. This goes back to our first videos. You might want to watch our first videos again from our course for the second exam, because it's a stem cell biology story where this is just normal muscle differentiation. And what happens in muscles is that, this is a figure from chapter 18, these are stem cells. And when stimulated, they will go through the cell cycle. So we can do our first drawing. Well, no, we're not going to do our first drawing quite yet. We're going to come back to this stem cell, and we'll say we'll do our first drawing, because they can be stimulated to go through the cell cycle. So right now, it's likely that all of your stem cells are sitting at the G1 to S transition, and you would call that G0 as if they're going on a detour, but they're not. They're just waiting at the G1 to S transition, and there's some rate limiting step that is preventing them from going through cell cycle, and they get stimulated when we need more muscle. The muscle fiber are differentiated cells, and in general, differentiated cells have permanently left the cell cycle, like the cells in our brain. We're not going to get more brain cells in this lifetime, like that. Well, those cells are also arrested at G1 transition, and we say they're at G0. They're not going to divide again. These muscle cells can't divide either. These are the muscle fibers that can contract and move our limbs and body around. They are done dividing, and they have limited amounts of growth they can do. They can grow, those tissues can grow some, but in general, when we want more tissues, we're going to produce more. And so these satellite stem cells need to be present all the way through our life, like that, okay? So the book goes through this and is just much less important, but it's good to hear that there are stem cells and this potential for new growth or regenerative growth in other tissues. So, for example, the liver can regrow. If you remove a big chunk of the liver, it can regrow. And that's different than some of our other internal organs. For example, our kidneys cannot heal. Our kidneys cannot regrow. That's why so many people have kidney disease. They have to go on dialysis. They have to desperately seek kidney transplant. And so the book talks about this, and that's good. That's good to spread your learning out, know a little bit more about it. Maybe there would be 2 points in the exam about one of these, but it certainly wouldn't be about the details of these different types of cells, and no one should learn those words. These are cells in our blood, so they're largely cells of the immune system and red blood cells. And this is not a course in immunology. So something's happening here, and that's going to be a feature of our first drawing, but we'll draw it when we get more information going. But you can see there's a kind of funny arrows here, and we'll learn more of that, and a kind of, I think, a much better way to draw that to represent what's going on. There it is there too. And here's our tissues in our digestive tract, and remember we learned something about that. We learned that there's that KIT receptor, and that the KIT receptor is a receptor tyrosine kinase, and it could be mutated, and it could cause uncontrolled proliferation in tumors in our digestive tract, and we learned, and so examples of that, and the increased mitotic index that happens in those tumors, and we saw examples of that a little bit, talking about mitotic index both in humans, and also we had examples of research articles showing the prognosis of dogs with certain cancers in that area. Yep. And so when we're talking about these stem cells, so adult stem cells, there are two things you want to say about them. They can make some specific cell types. So those muscle-like satellite stem cells have the ability to make muscle, but nothing else. The liver cells can make more liver, but nothing else. So they have a limited capacity of what they can make, of what they can become. And so one part of that limitation is they've already done some DNA methylation. And we've talked about that back at the start of our course, and we had questions about it in our first exam. And so, for example, in the muscle satellite stem cell, has it added methyl groups to the DNA for the myogenin gene? No. But how about the liver? How about those liver cells? Have they? Probably. They have a limited capacity to make cell types, and in the process of their development, they probably started proliferating some of the genes that they are never going to make. And so that's a little bit new to us. We learned that when a cell differentiates, it really is going to add methyl groups to lots of genes that it's never going to use. So when the cells differentiate as muscle, they are really going to be muscle cells. So that's a key feature of differentiation, is the cell methylates lots of genes it's never going to use. Here we're saying that these adult stem cells have really likely done some DNA methylation already. They've already limited what they can become. And I'm saying probably because that's a topic that's just, currently under research. But, logically, sure, you bet. The adult cells have already limited their capacity to form a variety of different cell types. And it's an important feature of them, okay? So the next thing we want to talk about is it comes up here, and it'll come up in stories of stem cells, and again, even if you're talking about plants. And that is that the stem cell, whatever it is, liver, muscle, etc., skin, intestine, and so forth, it has cells around it. It has neighboring cells. And those neighboring cells are actually releasing signaling factors to regulate the stem cell, to kind of basically keep it maintaining a stem cell status. And so these would be source cells that are making a secreted signaling factor, and then the stem cell must have receptors for those molecules. And people refer to that as a microenvironment, that if you take this cell away from this microenvironment, it will stop being the stem cell. And they also call it a niche. And a good place for us to think about that is in our bone marrow. So I haven't mentioned that yet, but our bone marrow also includes adult stem cells that are producing, well, I guess I mentioned it when I showed that we don't need to learn the terms of the immune system cells in our blood and our red blood cells. That was showing what's happening in the bone marrow. There's adult stem cell there, and they're dividing. Okay, and they're called hematopoietic stem cells, and they have a microenvironment. So there's just an article about that, and here's just an information. Bone marrow stem cell signaling is regulated by a complex interplay of factors. and look at what some of the signaling molecules are for our bone marrow stem cell. So we learned briefly about Notch in chapter 16. We learned briefly about Wnt. We learned briefly, maybe we didn't learn about sonic hedgehog. We learned briefly about TGF-beta. Those are the receptors that activate transcription factors called SMADs. And Wnt stabilizes beta-catenin, so we've learned a fair amount about that. It's come up a couple of times. And Notch makes the intercellular domain where the Notch receptor gets cut, and this becomes a transcription factor. So remember, we learned about that. Then down here, and BMP is another one that uses SMADs. It's a member of this big superfamily called TGF-beta. Then here's FGF, so that stands for fibroblast growth factor. No big deal, but it's a receptor tyrosine kinase. So it's in that family of 58 members that includes PDGF. So when you're pointing to this, it's not PDGF, but it's something that's in that same category. And then this one is stem cell factor. We called it the KIT receptor. And so you see, we learned about these, and they can be in a variety of different settings, and each of these also contribute to this fairly complicated environment in the bone marrow, and here's a picture of it. So I'll just draw these circles here. There's a hematopoietic stem cell. See, it's showing hematopoietic stem cells in this microenvironment and some of the different relationships they have with the other cells in the bone marrow, and even including, cells that are connected to the circulatory system, because of course we have blood vessels going into that bone marrow microenvironment. And again, we don't need to dive into this detail, but we can know that the stem cell in the muscle also has neighboring cells that are secreting factors and helping it maintain its stem cell status. Okay, so something that scientists realized they could do is they could take some of those stem cells out, and they could create the microenvironment in a dish by using purified signaling factors. It's not really the neighboring cells that are playing the role. It's what those cells are releasing into, the extracellular environment. So those signaling molecules could be used in a purified manner and just added to cells in a dish, stem cells in a dish, and you could maintain them in a dish. So we could take this and put it in a dish, okay? And when it's growing in a dish, we can study it. And so this is going to be our first drawing, and I think I'll show you this first. So we can learn in a dish that stem cells have really four things that they could do. So one is they could wait. So that's what's happening in our bodies right now. The stem cells are waiting, and they're at the G1 to S transition, and they could receive a signal, and it could be an extracellular signal like a growth factor, right? Like PDGF. So that fits in here. Of course, PDGF is, limited to the skin, like that. So that's the first thing they could do. Then they're waiting, and the next thing is they could start to divide. They could divide and go through, the cell cycle. Then another thing they could do is they could differentiate. They could just stop being a stem cell and turn into some type of cell like a muscle. That's a possibility. In principle, that's a possibility. Another thing they could do is they could go through programmed cell death. That's generally what happens to them. These are generally what happen to them if you take them away from the microenvironment. So if you put a stem cell by itself, it generally either differentiates or dies. And scientists in research labs are doing experiments on those and different options like that. So here's our first drawing, and maybe I'll skip over to my little drawing pad to draw this out. So we're going to use an example muscle stem cell. And there's our muscle stem cell. We draw a circle on it. And I just use an S to be a stem cell. And this is really a general drawing of stem cell. And the first thing to say is when it divides, and I said in their book they don't use a very sensible drawing to show it dividing. When it divides into two cells. And a really important concept, and the fundamental reason why these are called stem cells, is that is said to be self-renewing. So it divides into two cells, and one continues to be a stem cell for our whole life. When as it divides, it just makes a stem cell. And our example is a muscle satellite stem cell, so it's going to be dividing like this each time it divides. It makes two cells, and one of them is the stem cell, so self-renewing, and the other is something else. And the other is, we can just call, they usually have specific words, but we can just call it a precursor. And this precursor, these precursor cells to get produced, they can divide, and when they divide, they just make more precursors. And in the case of the satellite stem cell, those precursors start to change, and they become myoblasts, muscle precursor cells. And they're going to eventually express MyoD and differentiate into muscle cells. So this is a nice drawing of what happens there, muscle cells. And as long as the stem cell, the satellite muscle stem cell, is stimulated to divide and go through the cell cycle, it'll just be producing more and more of these precursors and more muscle tissue. Yay. Okay? So these are precursors. Then the next thing to mention is that people generally refer to this as an asymmetric division. So we have these cells dividing asymmetrically, unequally. And that's a key thing. And we'll see later in another drawing, or maybe we'll add that next drawing right here too. But this is asymmetric cell division. And that's that, okay? And so then I'm just going to erase this and add to this that we can add those numbered things, right? So we can add, if you have some good space, or you might just want to repeat it in a little second drawing as part of this one, is that these are the things the cell can do, right? The stem cell can pause. That's what's happening here. Then it can go through start. It can go through the cell cycle progression, and go through phase, divide, and make two cells. Great, okay? Self-renewing stem cells. Then another thing it could do is just be here, and if there's not the right microenvironment, it could just differentiate. And if it differentiates, it permanently leaves the cell cycle. It's done dividing. And then the fourth thing is it could just go through programmed cell death. It could be like, yeah, this is not working out. And stem cells are very poised to go through programmed cell death if the conditions are not right. That's the original reason that people understood that there are these microenvironments. The stem cell requires some help in being confident that it should not die, because it's a dangerous cell. It's a cell that we keep in our body throughout our whole long lives, ready to divide. That means it's a lot like a cancer cell. And so we want it to be very sensitive to the conditions and not just be rogue and go on its own and say, I'm going to divide, but really be sensitive. And if there's any issue about not receiving the right cells from the right signals from the microenvironment, the cell should just go through programmed cell death. So these are these other neighboring cells in the microenvironment, okay? So let's just put on here too, in this same drawing, or maybe we can consider this our second drawing, no, we'll make a second drawing in a little bit. So this will be kind of our first drawing, which is about adult stem cells and what adult stem cells can do. And as our example, we use the muscle. Can't make every type of cell. It has limited potential, but this is in general what stem cells can do. All right, great. So that's that, and our first drawing. And maybe you have it in two parts. Okay, great. So the next is going to be embryonic stem cells. So these are other natural types of cells. And so this is always surprising to share with students, and that is that embryonic stem cells are only found in mammals. There's no such thing as an embryonic stem cell in a frog, a reptile, a fish, anything else. They're only found in mammals. Those early embryos of those other organisms, non-mammalian animals, they just are doing something very different. And only found in mammals very early in pregnancy. In the first, in humans, only the first 7 to 10 days of pregnancy can you find embryonic stem cells. They're very early. And each embryonic cell during this period is a stem cell. Okay? And they have unrestricted potential. They can make the entire organism. And a good example of that happening is the example of identical twins. Identical twins have the exact same genomes from the two parents at this point. The identical twins are where one embryo splits in two during the first 7 to 10 days of embryonic development, very early in the pregnancy, and that shows that each of the cells has unrestricted potential. You could get identical twins, identical triplets, identical quadruplets. They're from one very early embryo that splits into 2 or 3 or 4 embryos. Fraternal twins are just two totally different siblings, like normal siblings are. They were produced by 2 separate eggs and 2 separate sperm. So what happens with them is that mom ovulates 2 eggs in one cycle, and both of them get fertilized, of course, by different sperm. And so we have really 2 siblings. They could be a boy and a girl. Identical twins are always going to be the same sex. Okay? So here's an image of that. So just, of course, from another book, here is the ovary, and an egg is ovulated, and it moves down the fallopian tubes. And as it's moving down the fallopian tubes is the time when it could get fertilized. That's why there's a range of time in the length of time that the early embryo is an embryonic embryo, is because there's different times when it gets fertilized as it goes down here. As it's moving down here, it's going to get to the uterus, and it's going to implant in the uterine wall. And so when cells are embryonic stem cells, they're during a time that people refer to as the pre-implantation time. And so this is called a pre-implantation embryo or blastocyst. We don't need to learn those terms. We just need to learn that term pre-implantation. They mean during this time after ovulation, when fertilization takes place over a period of potentially several different days and before implantation occurs. Here's embryo implanted, and these are really rapidly becoming no longer embryonic stem cells. And later in the embryo, we'll start to see the first examples of our adult stem cells, right? Okay, so here's what we want to do is we want to do a second drawing now, and that's our first drawing, and our first drawing, remember, says asymmetric cell division, okay? So our second drawing is going to be pretty simple, but it's pretty important, and then we're going to draw now an embryonic stem cell. And when it divides, right, they all have full potential, so it's going to divide over and over again, like that. And you can see they're just making more and more embryonic stem cells. And this is called symmetric cell division. So equal. So the embryonic stem cells and adult stem cells are distinguished by that, right? So think about this. I might have to scroll back to get to this image. And our second drawing is that simple, is to show that distinction, that embryonic stem cells divide symmetrically. They again, and we don't need to add this to the drawing, but they can wait. That's the first thing. They can wait at the G1 to S transition. The second thing is they can go through start. The third thing is they could differentiate. The fourth thing is they could die. So they have the same, kind of potential features. Another thing, and you could probably just have this on, just on your drawing, is they have, sort of, um, like we could call it like small amounts of DNA methylation. And that's just simply logical, right? They can make any cell type, so they're not going to have their. Does a embryonic stem cell have its myogenin gene methylated? The muscle genes methylated? No way, right? It can make muscle. It can make anything. So very little DNA methylation, and so that's just an important, simple distinction between embryonic stem cells. Let me just scroll back to this to show you this idea. So see these guys in the dish? So think about this. If you have your adult stem cells in a dish, you're not going to get more. They don't increase the numbers, because when they divide and make a precursor, and the precursor is not a stem cell. In contrast, if we get an embryonic stem cell in a dish, if we add the right factors to maintain its stem cell status, right, we can expand the cell numbers dramatically. We can actually grow this for years because we keep the right signaling molecules in place. It's a stem cell microenvironment. They maintain the stem cell status. They're just not going to do anything. They're not going to differentiate. They're not going to die. They're just going to plug away, making embryonic stem cells. So we can increase that number over a long time. The cells are fine. And at any time, when we want to do an experiment, we can just change the microenvironment. So change the signaling factors, and that can lead us to get the cells to stimulate. I don't know why I managed to move that around like that. That was kind of weird. Maintain stem cell status until we want to do an experiment. We can't get that from adult stem cells. That's a big reason why scientists are, two big reasons why they're really fascinated by embryonic stem cells, is they can make any cell type, very little DNA methylation, and you can expand their population and just get lots and lots of them, do lots of experiments. Okay, so here's just an image, no big deal, just because there are some folks that have identical twins in their family or their friends, and this just shows that when you are identical twin, they'll say, oh, we shared the same sac, or we were in two different amniotic sacs during delivery, because there are just slight differences in the timing when the embryo splits, and it affects the production of the placenta and the amniotic sac, like that. Okay, so here's a pre-implantation embryo, and they're just taking the cells out and they're putting them in the right setting, the right signaling factors to maintain them in a dish, and then they can remove the signaling factors or alter them and encourage the cells to differentiate in different cell types. So it's a figure from your book, because stem cell biologists are very excited about the possibility of, could we make some new blood cells for somebody who's lacking in blood cells? Could we make some new muscles for somebody that's injured, or somebody who's had a heart attack? Could we make some new nerve cells for an older person who's suffering from dementia or Alzheimer's? Could we replace some of those cells? Could we help somebody who's got a spinal cord injury from a car accident, like that? So lots of focus on stem cell biology. For us, it's just about, well, they're stem cells and they divide symmetrically and they have very little differentiation. Just want to show you this. We really don't focus on the scientists, but this was a scientist, Gail Martin, who provided a lot of really significant early work in culturing embryonic stem cells, right? Can't culture adult stem cells for very long because they don't divide, but you can culture those, and so she worked out the conditions for a microenvironment to grow. I wanted to show her because there was another scientist who was like in the generation before Gail Martin, who was in Maine and worked up at Jackson Labs, that's right next to Bar Harbor out on Mount Desert Island by Acadia Park. And I think he's probably the most significant scientist ever in Maine. Yeah, because he did some very early work, and it's just very, very early in the 1950s and probably 60s too. And there's a picture of him just to give a Mainer some credit. You don't get to see very many Mainers in science textbook, and Leroy was a pretty good one. And so, again, same idea. One thing that he and Gail Martin struggled with is in these cultures, the cells could do these things. And if they didn't get the conditions right, the cells would just go through programmed cell death, or they would differentiate. And I think, believe it or not, I think that's the reason why our textbook author chose to put stem cells and programmed cell death in the same chapter is just this experimental reality of you want to grow stem cells in a dish, well, you have to keep them from dying, right? Because they're scared that they're messing up, or just differentiating, being like, yeah, no, we should just finish up. Yep. So there's a lot of work, and when the cells differentiate down these different pathways, it's because we've shifted, not just simply removed the different signaling factors that we added to the dish. So, we didn't just remove, say, the Wnt, and inactivate beta-catenin, but we actually can change this around to encourage those embryonic stem cells to go down these pathways. So that's a huge reason why there's lots of funding in these receptor functions in our bodies. Is there's so much investment and research funding in these topics that will lead to regenerative medicine. So here's another piece I want to mention, and it goes like this. That to get these cells to differentiate, you can try to do this by just changing the signaling factors, but there's something else that's playing a role in what these cells choose to become. And what that something else is, around all of our cells, so here's some cells in our body, around all of our cells, there's a secreted extracellular material called the extracellular matrix. And the extracellular matrix is made up of polymers, long chains, so they make mesh. The extracellular matrix is really important in cartilage. The tough parts of our body that have cartilage holding things together. But it's always there, and it's full of lots of information. Importantly, an extracellular matrix is not diffusing. It's like a mesh, like a net. And it includes long polymers of protein. This is where collagen is located. That collagen that likely know is part of cartilage. They're long polymers of sugars, so polysaccharides. And that is something that's also part of this stem cell niche or microenvironment. And we aren't going to dwell on that because we haven't emphasized that in our course. We haven't learned a lot about it. We have learned a lot about those signaling factors, right? Factors like PDGF and all the other ones that we were learning about in Chapter 16. But I just want to mention this because this plays a big role, and I just want to describe and show you an example of this, okay? So it turns out that if you just take these stem cells and you try to get them to differentiate into tissues, they can make muscles, but they aren't very well organized. They just don't really contribute and participate. And one of the reasons is because they don't have that extracellular matrix. So one thing that scientists did years ago is they would take an organ, like a kidney, and they would remove the cells, and they call that decellularization, to remove the cells, and they would be left with this mesh. So they would kill the cells in some way, with some treatment. And so then they would have this, and it would still be the shape of a kidney, but it would just be that extracellular mesh. And it turned out that you could put embryonic stem cells into that mesh, and they'll make a kidney. They'll just repopulate that kidney. So they found quite a while ago that these embryonic stem cells need that mesh, need that information too. Now, this isn't very helpful for regenerative medicine because you have to ruin a kidney to get a kidney. But it does point to more than just these signaling molecules are needed for making a new kidney. And so here's the next thing, and that is, just an image from an intro bio textbook about this extracellular matrix. The next thing is now some researchers are using 3D printers to print the mesh and then get stem cells to grow and form tissues. And this is a real thing, and it's very modern research, but people have been working on it for a while and trying to increase their ability to do things. And what they do is the printer has, the inks, right, to print, and normally on your printer at home, the inks are inks that go on the paper. And then in a 3D printer, the inks are like liquid plastics, or some liquid material. Here, the inks are going to be like parts of the extracellular matrix. Plus, one of the inks is going to be embryonic stem cells. And scientists are really excited about that because they're really starting to make some really good advances. And so that's just a good example to be aware of for us, that the extracellular matrix, so that's just a term, and it could be a 2-point question about, what did we learn about the extracellular matrix and stem cells? And we learned that it's an important part of guiding the stem cells toward differentiation of what to become and to properly organize things like a kidney. So there's just some work they're doing on that, just an example of that, okay? So these embryonic stem cells, we also have this story of the DNA methylation. And I already mentioned it, and I'll just mention it again, that embryonic stem cells tend to have reduced DNA methylation, like that, okay? All right, so now we're going to talk about induced pluripotent stem cells. And this fits into our course very, very nicely. We can really dive into this, and we'll try to do it quickly and efficiently and build this framework for understanding these. So these are not natural. And it's a story where scientists discovered they could take any cell from your body, and the example we'll pick is a muscle cell. And they could do work in lab to convert it to a stem cell, and a stem cell that's like an embryonic stem cell, a stem cell that could make anything. It could make all the different cell types. And they're excited about that because it has a lot of relevance for medicine. Because if a person has a damaged kidney, maybe we could take that person's cells, turn them into these stem cells in a dish, and grow them a new kidney. Would they reject that kidney? No. It's their kidney. And so that's really exciting, the idea that you could take a patient's own cells, make tissue that is the patient's tissue, and then put that tissue back into the cells. So they're called induced pluripotent stem cells, or IPCs. Like that, IPCs, induced pluripotent stem cells. And the pluripotent word is meant to indicate that they can make all the different cell types. So let's just think about two things right here, okay? And they are, what's it going to take to make an induced pluripotent stem cell out of a muscle cell? Really two things. It's differentiated. It's a differentiated cell, and this is something we've emphasized since day one. It's got a lot of DNA methylation, so we're going to have to demethylate lots of DNA, right? It's a muscle, so it's methylated genes it's not using. Is the insulin gene methylated? Almost for sure. If we want to make a stem cell that can make anything, is it going to be able to make a pancreas? It's got to. Does it need to be able to express insulin? So we need to demethylate the DNA. And right in here, this is the reason why we chose to focus on these differentiated cells and their methylation status, is because we really want to understand this idea of it's got all the genes, right? So this has, super importantly, this has genomic equivalence. Has all the genes, it's not using them all. It can't use them all. Some have been methylated, demethylate them. And then the second thing is go through start. Go through the G1 to S transition, re-enter the cell cycle, because that's a feature of muscle. They're not going to divide. Can you take a brain cells and turn it into an induced pluripotent stem cell? Yes. Demethylate lots of genes and get it to start to proliferate again, okay? So here are the two scientists that won Nobel Prizes again for it, several years ago. This guy did his work, especially early, and and was the first person to really show experimental evidence for genomic equivalence back in the early 1960s. Then this guy did all these experiments, and he's a molecular biologist in Japan, and he did all these experiments very recently, in the last 15, 20 years. He did all his work. And he had an idea. And his idea was, well, you say that, that this cell has genomic equivalence. Well, it has all the genes, and we've learned that muscle cells express muscle genes. The pancreas expresses pancreas genes, right? They've got all the genes, but they're expressing different sets of it. The stem cell must express some stem cell genes. There must be genes in here that are being regulated in the stem cell niche, in the stem cell microenvironment, right? There must be some signal transduction pathways activating gene expression of genes that are specific. And when the cell divides asymmetrically, it must be self-renewing because they maintain the expression of those specific genes. And then this becomes a precursor in the case of adult stem cells because these stop expressing those genes. So Yamanaka, he knew, I don't need to study the DNA, I need to study the RNA and proteins. And his lab started to compare the stem cell with the precursor cell, what genes expression are different. And they initially found about 20 that were seen to be significantly different, and eventually they got it to be, there are just these 4 genes. And if you take those 4 genes and express them in any cell, it'll turn into a stem cell, and that's what an induced pluripotent stem cell is. Whoop. See, where's that little name? So an induced pluripotent stem cell is a cell where you've misexpressed 4 genes. Okay, and so we're going to draw. Our final drawing is going to be those genes. Okay, and so let's just look because there'll be a list of them coming up. So there are just these 4 genes, and here they are. And they're all transcription factors. And just expressing those can turn any cell into a stem cell. So they're present in the cell, we just need to get them expressed. Okay? And they really fit into 2 categories. And remember what the categories are, because we already talked about them. One is, these are transcription factors that activate genes that include proteins for demethylating DNA. That's their big role. And the other category is c-MYC. Remember that cellular MYC? That means it's the normal MYC, but they're going to force the cells to make MYC. And think about this. They can make any cell go into the cell cycle. You can take your brain cell, your muscle cell, and they can force it to go into the cell cycle by overexpressing MYC. Just that. Do you want to think of cells when they're arrested at G1 to S transition as going on a big detour through G0? No. They're just waiting at a red light. And just think about how amazing it is that you could push them through that just by overexpressing that MYC, and how dangerous MYC is and how carefully a cell regulates it. It's really surprising, and it's kind of unbelievable that could get the job done. But that appears to be the case. And scientists are learning more about it and understanding it more and more. These cells have roles. It's a complicated role. I'll just show you this. Our book shows this. These are the 3, and they have a complicated interaction where the transcription factors are activating and repressing each other. And there are a bunch of different target genes that they are regulating, and they also are involved in that cell cycle progression. So it turns out not quite as simple as, oh, it's just MYC, So that's the first way I expressed it, but it's not quite that simple. But it's startling what MYC can do to drive the cell. And so that's, again, what we want to know is that, okay? So now we just want to just draw a picture for our final drawing and show what the scientists did to get it started. So we appreciate that. So let's get over here and make a drawing, because what they're going to do is that the stem cell it has genomic equivalence. So it has the MYC gene and the other 3 genes that are needed to be turned on, right? So it has genomic equivalence like that, okay? But the muscle cell, and we'll just use our muscle as our example, that has these genes, but it's not expressing them. And they're probably methylated, right? Because it's no longer a stem cell. So it still has them, but it's not methylated. So, and the experiment so far in trying to activate MYC and the other 3 in the cell, what they did is they built genes that are transgenes. So I guess I'm drawing this a little bit upside down from what we usually do. And so they would take a MYC gene and they would just put on a different regulatory region in a test tube that they could control and force it to be activated. So it's the same MYC gene transcription unit that we're used to, but they're just not going to use the one that's in the muscle cell. They're going to take one from outside and build it in a test tube. And they're going to build 1 transgene, and 2, and 3, and like that, and 4 in the test tube. So they're going to build them in a test tube, and that can be your drawing for this one. And that just allows them to put them in and control expression. And and we're not too worried about being able to label these. They do have sort of curious names, but in each case, we would just add a regulatory region that we could control and get to be activated. So even though the muscle cell already has these genes, the way to get this done is to put on, in a test tube, some regulatory region that we can control when we put them in the muscle cell. And then we're going to get them all in the muscle cell as new genes, and we're going to activate them and activate their expression. And they're all transcription factors. So all of them will make proteins that will go on to activate a bunch of target genes. So we're not surprised that if you have just a very few genes that can make a big effect, it's a good chance they're transcription factors, because they could turn on a bunch of different target genes and get a big change in the cell. So that's our third drawing, I think, that we'll talk about saying, and it's just showing that, just the reality of the situation. It's like it's called misexpression. So we're going to misexpress, we're going to force the expression of these guys. And that's sufficient to turn any cell, any just normal cell in your body, into a stem cell. They could take a skin cell, a brain cell, a muscle cell, kidney cell, a liver cell, and so forth, and turn it into a stem cell with those. So it's just fascinating, and it really is a way of seeing, wow, our course really helps us appreciate that that's how you could get that experiment done. Great. And so the book describes a little bit about the complexity of the story, and it's complex, but we're just trying to establish a framework. And again, there are the three proteins, and you could learn those terms. Another kind of cute thing is that the scientist was named Yamanaka, so a lot of people call them Yamanaka factors, so you could refer to them as Yamanaka factors, or this just stands for OCT, SOX, KLF, and MYC. And so great, what we're especially focused on is they're all transcription factors, and they're already in the cell, but to get them going, we're going to, force their expression. And this is the book diving into some details about that, because it's, current research that scientists are very excited about. So we've got to get it in there. And so again, back to this thing is they're going to reduce the DNA methylation, and they're going to drive the cell through the cell cycle. And so here's just a different image from a review showing that activation and that pathway, because we can make the stem cells and then we can maintain them. We can expand the population in a dish, right, by keeping the right signaling molecules in the dish. Then we can change the mixture of the signaling molecules, maybe add some extracellular matrix, and we can make any tissue we want. That's the goal. And that's where scientists are at right now. So it's pretty cool because it really is something that is justifiable to talk about in this course, that we really set the stage up to be able to talk about this in this framework and to understand really what the scientists are working with and struggling with. So it's all very cool, and we're all set with this. So we're going to do those drawings as part of this, and just try to get in the habit of trying to draw it out, try to frame the story out as best we can in a memorable way. That's what important way that you can learn and then recall. You can recall the pictures. And my drawings, are very sloppy. They don't have to be fancy, but hopefully they'll be memorable. That's the whole point. You're not just kind of grinding out the assignment, but you're trying to make it a memorable experience. All right? Super duper.